Okay, so let's have a class. So some of you still looking for that's okay. We are going to move to the biochemistry reaction number 3 today. But first of all, we have to do the confirmation test of biochemical 2. Okay, so let's do the confirmation test. Number 1, what we did is catalyst test. Catalyst test, if you still remember, we have H2O2, then should become water and oxygen. If there's oxygen comes out, we call it a catalyst test positive. Therefore, we need you plates, is catalyst plates, have it out on your bench. Jess, can you go ahead to add H2O2? Right here is H2O2. Add onto it. And I will do right here, use the one as a demo. Okay, for H2O2. Remember, the plates, what you do confirmation test, is one side E. coli, another side is Staphylococcus aureus. So Jess is going to help you to do that. I'll just do a demo myself. Okay, when you add it, you should see it looks like this. So the bubble comes out on one side. Can you see it? The other side, there is no bubble. Okay, when the bubble comes out on one side, it is catalyst test positive. So which one? This is Staphylococcus aureus. This is E. coli. There is one side, there is a bubble comes out. You should know. It is Staphylococcus aureus. This bubble comes out. We say bubbling. Okay, that's catalyst test positive. Always remember. So, what we're going to do is that, Jess, you're doing that. Just a couple of drops is enough.
Yeah, it's working great.
Okay. Yeah, have you plates ready? Okay, I'm gonna give it to Corey. You're gonna help everybody else to add H2O2 onto the plates. Okay, that's your job. Add yourself, added H2O2 for everybody else. Don't use all of them. Yeah, just a tiny bit drop, press. Yeah, one side of each. Then you are wait there, you see the bubble comes out. Okay, go ahead to from the bench, number 1, and the side, go ahead to add it for everybody. Is that okay? That's good. So you guys can take a look. Okay, so we move on to the second test, which is oxidase test. Now remember, electron transfer from cytochroma C to following stuff, let's say oxygen, they need oxidase for electron transfer. So what we have is agar plates. On the left side, we inoculated the Neisseria. On the right side, we did the E. coli. And the test is also very simple. We are using is the oxidase test reagent. Is just like this. Okay, when you see the label, the oxidase test reagent, it's a very long name. So we're going to add a drop, is oxidase reagent. So I'm going to use this example to show you how to do. And then, Jess, can you go ahead to start?
Yeah, I think I got this one.
Yeah, Jess, go ahead to do oxidase add to every reagent, okay? So this is the same thing what I'm doing right here. We'll be add a couple of drop on each side. We will be stay around around about less than a minute, we're going to see the 30 seconds to see which side is showing you kind of like a purplish color. And you can see one side is already showed the purple. Is nice area. That part is already showed the purple color. So the same thing here. Ian, can you go ahead to drop on every plate, including yourself, for this half batch? So what you're going to see is, on the nice area part, you will see kind of like a purple color showing up. That means oxidase test positive. But be careful, this is you have to do is within 1 to 2 minutes. Because otherwise everything will become positive. The reason is it's going to expose to the air. Okay, so that's oxidase test. You can see very clear nice area is positive. Okay, this is number 3. They add the drop there. You can just wait and you can see the color later on. Okay, then take a picture. SIM test. Okay, so let's go these one by one. S sulfide. I is Indo. M is motility. Okay, I still have some left sample here, so let's talk about all these. Everybody look at this sample. I better say this is Citrobacter. What do you think? Sulfide positive, black colony, okay? So you record your own results, but I tell you one thing, Citrobacter is a very typical, is sulfide positive. It is black colony, black color, we can say. But why it is black? Because metallic ingredients will let sulfide reduce to H2S, and then metallic ingredients show you black color. Okay, now the motility, what do you think? Is that motility positive? It goes everywhere, is that right? And some of you did not show the black color. You can take a look. If it stay in the middle, it's negative. So I will say this one also motility positive. Now some of you, if you see your sample is right stay in the middle, then it's negative. We usually don't say positive or negative. They just say there's no motility or limited motility. Very possible. This possibility is the flagella is cover the whole surface. We call it a petrotrichus. Petrotrichus. And this is monotrichus. Okay. So that's, we talk about the motility and sulfide. Okay, how we test the Indo? And I need to find a reagent right here. How we test the Indo? Can somebody tell me how we test the Indo? Do you remember? Which reagent do we need? We don't have it?
Wait, what's in there? Let me see.
What is the Indo? I have it. What's the name? How we test the Indo, if you remember, we're going to add a 1 drop of reagent, 1 big drop. What's the name of it? Kovac. So I'm going to do this one test for you, okay? I may only have this one left. These reagents always shake first, then we're going to add 1 drop. No reaction, no color change. Who has E. coli? Give it to me. Just give it to me. Very good, E. coli. Let's see E. coli as that looks like. So this one you can see right in the middle. There is a line. So E. coli's motility is not very strong. Let's do this test. Indo test. Cup of drop. Do you see cherry red color there? How about this one? Nothing there. Can you see it? E. coli is Indo positive. And always remember, the easiest way to test E. coli is Indo test. It's Indo positive. Showed you color is cherry red. After you add a Kovac, it is cherry red. Always remember, and you can do the test by yourself. Don't use all of them, okay? You can do the test by yourself. Number 4. Phenylalanine deaminase test. What we should add? You remember? We need to add a 1 drop FeCl3, ferric chloride. If you see the color, which is showing you dark green color, it is positive. Let's do it. Okay, I assuming this is inoculated. I don't know what it is. Staphylococcus aureus, we'll see. Where is ferric chloride? I need to find the reagent, the ferric chloride. On the front of the desk, we can see it. I don't know whether I trusted this or trust the one I bought. Okay, we'll see. So add a couple. We'll see whether it is positive or not. I don't think none of them will be positive. Do you see it's positive? No, that's negative. You can go ahead to do. You can go ahead to add for them.
Yeah, they've got.
To see, you have E. coli and you have Staphylococcus aureus to see which one is positive. Maybe they're all negative. Okay, you may get up all negative results. Yes. Okay. Now, the last one. MRVP test. Okay, MRVP has E. coli. There is a 2 way to do, which is E. coli versus Enterobacter. How you do it? As a pair, is that right? And you also working with a person who is across the aisle. So which means 4 of you will be a group. Okay, you have E. coli. You have Enterobacter. This is tube number 1. This is tube number 2. And across your aisle, you have Enterobacter tube number 1 and Enterobacter tube number 2. What we do? One of the tube, you're going to add. Tube number 1, you add methyl red reagent. So methyl red reagent is just right here. It's so easy. Methyl red, it said just like this, you added it. So I'm gonna just do a test, okay? I don't know which what is this gentleman they did. It's supposed to be E. coli. I didn't see label very well, so I'm gonna add it. Did this show pink color or not? You can go to add. One of them is using methyl red. One of them. Don't use all of them. Did you see any change? If you see pink color comes out, it's positive. This one doesn't have pink color. So methyl red, if it's pink, it's positive. Then you have another tube. It's tube number 2. You add a VP test reagent 1 and 2. You need to add both. Okay, you can do this by yourself later on. I test 1 and 2. If it's pink, it is positive. Now, what we are testing for? Methyl red reagent we are testing during the fermentation. They generate acid and products. And the VP test we are testing neutral and products after fermentation. And obviously you know the results. E. coli, Enterobacter, they are a pair, their results are very interesting, MR and VP. E. coli is negative, negative, Enterobacter is positive and positive. IMVC test, E. coli is positive. It should be this way. E. coli is positive, Enterobacter negative, E. coli VP negative, Enterobacter is positive. That's going to be the results. Yes, you add the methyl red first. Another person, not everything, just not every tube.
Right.
Then you add reagent. Reagent B is for methyl red. You can do that later on. Okay? So this is the one you do it a little bit later. Okay, we're gonna move on to today's lab. What is that? It's so simple. Okay, today's work. Everybody will grab 3 plates. Number 1, starch agar. Number 2, skim milk agar. Yeah, somebody already talked about the skim milk. That's good. That's a good point. Split brew agar. Okay, what will I test for? The work is very simple. At the back of the agar, split them onto 2. For starch agar, on the left side, inoculate Bacillus subtilis. Right side, inoculate E. coli. Skim milk agar, same thing. Left side, streak inoculate Bacillus subtilis. Right side, inoculate E. coli. For split blue agar, left side, inoculate Staphylococcus aureus. Right side, inoculate E. coli. Now do not forget, very important, is label your batch number, initial, and date. Okay? Label your batch number, initial, and date. Batch number, initial, and date. So the work is very simple. We do the incubation afterwards. We look at whether there is a transparent zone surrounded the colony. We want to talk about all these one by one. Starch. Why we want to test the starch? Bacteria use glucose, you all know that. And sometimes bacteria can use a higher level sugar, which is a disugar, disaccharides. Starch is one of that. What's the components of starch? Amylopectin and amylose, is that right? How are they connected? 1,2 or 1,6 glycosidic bonds. So one is like a circle, another one is like a straight. So you need to know that. Which enzyme is for bacteria to break down starch? Amylase, yes. So starch agar we test. Which bacteria has amylase? Now, how do we know it's amylase? How do we know there's no amylase? And I just said we see transparent zone, but probably not at this moment. How is it? How is it? Which reagent reacted with starch? Is iodine. If iodine reacted with starch, it shows brownish color. So, after incubation, 35 degrees Celsius, 48 hours, we need to dump iodine. If you see it shows brownish color, what this means? The starch is still there. So, no amylase. If you see no brownish color, the amylase is positive. Okay, so twist your mind a little bit. After incubation, we're going to dump iodine. It's the same as gram iodine. If you see a brownish color, which means starch is still there, because starch is reacted with iodine, showing brownish color. If you see the brownish color is gone, there is amylase, because amylase has been hydrolyzed, starch become glucose. Okay, that's very simple. Okay, skim milk agar, what we are test for? You should know there is 3 different grades of the milk. Do you know whole milk? 8% fat. And 2% fat reduced milk. We call it a reduced milk. What is skim milk? 0% fat. Okay, other than fat, inside of skim milk, which one is a major component? Of course, water is a major component. Other than that, what is that? What it is? Protein, is that right? Okay, protein. What is the major protein in milk? What? Lactose is a protein or sugar? Okay, they're not lactose. They're called casein, is that right? So sometimes the bacteria can use protein if they have a protease. And in this case, it's casein. So how do we know it is positive? That's very simple. If you see transparent zone. Okay, so this is a test of whether bacteria have caseins or have protease. Okay, the last one, sprit brew agar. This is the one bacteria sometimes use fatty acids. Which enzyme to break down fatty acids? Lipase. Okay, so this is test of whether bacteria has lipase. Some of the bacteria has lipase. Let's say lactic acid bacteria. That's very important in the meat processing. Because we want to control the amount, the ratio between lean and the fat. We don't want the fat is too much. We don't want the lean too much sometimes. We have to be a balance. Because when we talk about the meat, we talk about juiciness, flavor, and the tenderness. And the amount of the fat will be affect that. So that's why sometimes lactic acid bacteria has lipase. We don't have lactic acid bacteria, we have Staphylococcus and the E. coli. Same thing. If you see transparent zone after incubation, which means it's lipase test positive. Okay? So today's work, what we left? 3 plates everybody get. Starch agar, sprit brew agar, and egg and skim milk agar. Okay, let me show you all these. What it looks like. Sprit brew agar. We already said it's sprit brew. Of course it's brew. It's so simple. Which one is the skim milk agar? You tell me. Yeah, the milk. very white. Okay, so you don't have to label in there. You can go back to the bench to label. And this is very similar to our general agar. It's a starch agar. Okay, so it's very clear. I really like this test. It's very simple and straightforward, but tells us lots of information compared to all the other TDS work. Okay? Don't forget, if you can, don't forget to label your name, bench number. If you're confusing on the side, write an animal, write some marker there. Then make it easy to find it. Other than that, all these confirmation tests you want us to do, you go ahead to do. Look at here. Once it's more than 1 minute, what happened? They both become purple, blue color. So that's why I tell you, you have to do this is within a minute. Okay? And another thing I want to mention, VP test, it maybe take a little bit longer. So the VP and MR reagent, you could put on your rack, and we can look for next Tuesday because it took a couple of hours to react it sometimes. It really depends. Okay? So we can go ahead with it.